Good morning. Today I'll be talking about recombinant DNA technology, which is also called as rDNA technology. Okay, rDNA technology basically involves cloning. Now, what is cloning? Cloning involves something that you make it repeatedly. Okay, here we are going to clone our genes, and hence cloning is also called as DNA cloning or gene cloning, or it is called as molecular cloning. Okay, in layman's, if we try to understand what is cloning, you can take an example of a car. Okay, car has a model. For example, if you take I-20 as a model, every time I say the word I-20, every time you are going to make the same model of the car. Same is the case with cloning. If I give you a gene sequence with the help of a cloning process, every time you are going to have the same gene copy. Okay. So I will give you an overall of the cloning process. In cloning process, you select a gene which is a gene of your interest. That gene is inserted into a vector which acts as a vehicle. That vector is then inserted into a bacteria which acts as a host. That bacteria is allowed to replicate, and we get ample amount of that gene which we had inserted into the vector. So this is the big picture of the entire cloning process. Now, just like I mentioned, you need three different things to carry out the cloning process. First is your gene of interest. Second is you select a proper vector for the process, and third is you select a proper host for the process. Okay. Now here we'll take an example of a gene called as insulin gene. Okay. Now this insulin gene is going to code for a protein which is going to get synthesized. Okay. Now to synthesize this particular protein, the next step is we should finalize a particular process. Okay. Now this process depends on the host as well as the vector that you use. Now you can say that this process can be carried out in vitro using PCR technique, that is polymerase chain reaction technique, and this can be carried out in the test tube as well. But the drawback of PCR is whenever you synthesize genes in PCR, that is only the gene that you are working with. Okay, that gene will not be able to prepare proteins because that gene will not have promoter or maybe a start codon or a stop codon for translation and transcription. So if your aim is to produce protein out of that particular gene, your PCR technique is not going to work. So for this you need cloning technique which will get you protein as your end product. Okay. So once you have finalized the process that you are going to use for cloning, next important thing that you need is your host, and after that you are going to get your product. So if I summarize this, you need to identify your gene, you need to identify your process, and after you have finalized your process, you need to identify the process by which you are going to extract your product from that particular host bacteria. So this is the overall summary of the recombinant DNA technology. Here we have isolated the gene which we are interested in. That gene is then inserted into a plasmid. That plasmid is taken and then inserted into a bacteria which is also called as the host. This host then replicates, and because of the replication of the bacteria, ultimately your DNA that is inserted in the plasmid is also getting replicated, and then this recombinant DNA can be further used for various applications. Okay. So the process in which your foreign gene is inserted into the plasmid and you get a plasmid with that foreign DNA, that process is called as recombinant DNA technology. Okay, and when you get the replicas of that particular DNA when the host multiplies. That particular process is called as cloning technique. So recombinant DNA is a part of the